Okay, you guys, here we are in the hotel room, and I'm going to make a presentation on Chapter 7. So here we go. Let's see how this thing works. So, Chapter 7 is about membranes. Yeah, looks like you can see that. So, we're going to take a peek at membranes. I wanted to remind you, of course, what they are. So, we can do our standard little phospholipid bilayer thing. We've done this before. Remember, polar heads, nonpolar tails. And I wanted to remind you also that we've already identified big proteins that sit in the middle of a phospholipid bilayer. So you can see this kind of action going on here. So this is the big protein. Remember also that this part in the middle of the protein, this part right here, had to be hydrophobic. So that's our hydrophobic, we call it a domain of a protein that goes all the way through. So what we want to do is actually give more language to this thing and give a name to it. So this type of protein that goes all the way through a membrane is what we call an integral protein. And there we go, new term. So the integral protein, let's see, sorry, I had to double check to make sure we're recording. So the integral protein is one that goes all the way through the membrane. There's another kind out there too, and that would be called a peripheral protein. So this is one that is pretty much attached to a membrane, but not really part of the membrane. So I guess over here, maybe I ought to draw a peripheral protein. So here's a section of phospholipid bilayer, of course, as we always draw. And then I'll get a peripheral protein out here to take a peek at. Let's see. Should we get that thing off of there? Okay, good. All right. So let's make a peripheral protein right here. That's the peripheral protein. Of course, I think we can identify a couple of other features. The way I've drawn it, this is extracellular space, and then this, of course, is this is cytoplasm. You know, cytoplasm intracellular. That's where this lives. So there we have it. So that's what peripheral proteins are. I'm not going to say anything else about peripheral proteins. I'm going to pick up here with integral proteins. So as I go down the board here, this is going to be all about integral proteins. So we talked about the hydrophobic domain, that it's penetrating the deal. There's actually a couple ways that integral proteins can be. The first type, of which again, this is one, is what we will call a transmembrane protein. So we're going to have layers of terminology to break these down. This one goes all the way across, like transcontinental, that's from coast to coast. This thing goes from the from the extracellular to intracellular. I'll draw another one here that you haven't seen yet, and this one would be what we call non-transmembrane. So other, I guess I could say non-transmembrane. There may be a name for this, but your textbook doesn't have that name. And I'll just draw a little picture of this. So here's a little bit of phospholipid bilayer. Then here's, I'll continue this way. And then I'll leave a gap here. I'll get out a marker and draw a transmembrane protein that doesn't go all the way through the membrane. So this guy still is an integral protein because it's in the membrane. It's the type that is not trans because it doesn't go all the way. This one right here only goes part way through the membrane. All these things are important. There's different kinds. All right, so let's pick up with the next thing on our plate, and that would be the types of proteins now, the types of integral proteins. So here's kind of the size and how they relate to the membrane. There's actually more specific types of transmembrane proteins. This is big stuff. So types of it's a big title right there. Okay, so you can see the board. So let's go with the first one. The first one is a transport protein. This will be huge today. We're going to talk a lot about transport proteins. So this is big stuff. It's so big that right now I'm going to take the time to put a nice star by it. This is big time stuff for today. That should catch your attention is what I'm saying. So transport protein, and in transport proteins, these go all the way through a membrane, and we're going to see two different kinds. We're going to see, well, let's just give a picture of a transport protein. We're going to see channel proteins. Channels will look like this. Phospholipid bilayer, as always. Transmembrane protein. Should probably keep up with the colors here. Transmembrane protein coming all the way through. I'm going to draw this so you can see that there's a hole that goes all the way through it. It's like a bead or something, and the hole in the bead goes through the center. And of course, let's draw a little bit more phospholipid bilayer to complete the deal. Excellent. So there we have a channel protein, and you can see that something, maybe some kind of a solute, could go traveling right through that. Let's do that. So let's say that we've got some substances here. That substance could go all the way through, and I'll put several of them up here. That's going to be important actually later on. But this shows that we've got these little blue solutes up here, and here is this bead-like structure or donut-like structure, which is a transport protein, a type of transmembrane protein. And through this thing, we've got the thing just flowing. All right, so that is that. Let's keep going now. The next thing is that I want to talk about for transport proteins is what we call pumps. Pumps. Now, pumps are going to be things that will require, oh, not pumps, I'm sorry. Pumps is later. There we go again. Carrier proteins is what we want to mention. Carriers. And carriers are going to be things that will require a shape change to get the job done. So let's draw a carrier right here. A little bit of phospholipid bilayer. And so here's this thing called a carrier protein. Change colors again. Let's make an orange one. So this thing goes all the way through, but you'll see that it has binding sites of sorts on the inside. And as drawn, this thing is open to the intracellular part, but it's not open to the extracellular. So this is how this thing works, or looks anyway. We might see that down in here would be solutes, little diamond-shaped solutes down here in the cytoplasm. Maybe there's one out here. And what this whole thing is going to have to do to get its job done is, in a later picture, the shape change will reverse. Let's see. Try to get some of my plasma there. And okay, here we go. I'm sure if you guys can see that okay. So now the thing is going to shape change and open up the other way. And of course, when it does that, I should have shown that we had a couple of these things bind here. So these things actually, these solute molecules bind to this. Well, I'm actually already leading the way. We're talking about substances crossing the membrane. But now these things are going to come out and get on the other side. So there's the shape change. That's what I really want you to see, is that, let's write it down, shape change. Okay, all right, so there's our transmembrane proteins. There's two categories that we have. Let's continue this list now. The next type of thing that we could have there would be called an enzyme. Now, enzymes are going to be special proteins that catalyze biochemical reactions. Catalyze is speed up or make happen. We'll talk about this later. So catalyze biochemical reactions. I'm going to put here something to indicate to you that this is going to happen later. We actually will talk about this in another chapter. So you're just going to have to take it, face value that this is a type of transmembrane protein. The next type of transmembrane protein, the ones that go all the way through the membrane or partially through the membrane, the ones that go all the way through all the way through the membrane, transmembrane. The next type is ones that are used for signal transduction. Single transduction proteins. This list is straight out of your book. So these guys will bind molecules on one side, like the extracellular side. For example, hormone receptors that bind hormones outside of the cell. They'll bind hormones on the outside of the cell, and then this will cause a shape change event. Shape change that impacts the inside of the cell. Okay, so let's see if you guys can read that. I'm going to show you the whole board for a few seconds here, because it doesn't look like, yeah, I think you can read it. I can, I'm looking at the screen. So, signal transduction protein, something binds to the outside and in a receptor, and that causes the protein that's in the membrane to change the shape, and that in turn kind of chain reaction impacts the inside of the cell in some way. So a signal from the outside has been transmitted to the inside of the cell. That's the key for signal transduction. This is kind of an interesting part of biology and cell biology that we really won't go into very much. So that might be about it. Okay, now 4 and 5 really important. Cell to cell recognition proteins. So that's the next kind of transmembrane protein. And these are things that are often important for one type of cell to recognize another type of cell. Examples are, these are very common, they're important, I should say, in our immune system. So our immune system, big, huge, important thing. The big player of our immune system is called a white blood cell. Now, white blood cells are cells that, we're going to learn this term later today, but what they do for a living is white blood cells are what we call phagocytic. That's an A, phagocytic. This is going to happen later, but phagocytic cells gobble up other things, including other cells. So white blood cells are phagocytic, and what you need to have in cell-to-cell recognition, one thing you need to do is, if you're a cell that can gobble things up and destroy it, you can't gobble up and destroy the organism. So white blood cells like this are phagocytic, but if there are proteins on other cells that shouldn't be gobbled up, then that is cell-to-cell recognition. So this is kind of preventing the autoimmune phenomenon almost of cells eating cells that they shouldn't eat. So you're going to see what phagocytosis is a little bit later, but that one I wanted to point out is pretty important. And this is with proteins. It's kind of like the fingerprint of the cell on the outside, the proteins. You don't get much of a fingerprint from phospholipids, do you? Because they're just polar heads and non-polar tails. Not a lot of differentiation. But proteins, as we've talked, are incredibly variable molecules, cell-to-cell recognition. Okay, next one is what I'm going to call intercellular joining. You know, I want you guys to think about any kind of, organismal tissue that you've held. Think of like a liver, maybe you've held a beef liver or something like that, or a steak or something. The cells, the individual cells that make up that tissue are stuck together. And they're stuck together almost with rivets made out of proteins. In the protein category are transmembrane proteins called intercellular joining proteins. So there we go. So I want to share with you that both of these things here are involved in cell-to-cell. But this one's the robust one. They stick together really intensively. This one's short-term. Usually the cell-to-cell recognition phenomenon involves a short-term joining of cells. Alrighty, so that's great. Number 6, the last one, I can erase this board, is the attachment proteins for what we're going to describe as the cytoskeleton. So this is something that we're going to do later as well, we're going to do, this gets into the next chapter. I'll put a nice later here. So this outline might be a little bit frustrating because it's a bunch of names, but we don't really get to talk about a lot of this stuff in detail. Today, we are going to completely knock out. This is today. We are going to completely knock out this entire section. This is our mission to understand this in glorious detail today. All of this stuff, list and terms, concept. That's what this is for. So you'll obviously be memorizing the names of these kinds of things, but we talk about this in a later chapter. We talk about this one in a later chapter. We talk about this one in a later chapter, and we probably won't say much about, I'll go right later here. Later. We probably won't be talking much at all about signal transduction. So this is our goal for the day, to knock this out. Now, to do that, really what it comes down to is a concept that we call crossing the membrane. Let's see how these little erasers work. So going up to the top to erase. Not bad. So you guys know that I'm at a meeting. I'm in Layton, Utah, which is it's north of Salt Lake, and it's pretty amazing here. This is not my first presentation today. I gave another one to 150 people, and it was legendary. I mean, I knocked that thing way out of the park. It was great. So sorry if this one's not as good. This is kind of a weird environment for me, but I think we're pulling it off. So we're focusing on these transmembrane proteins. I'm going to go ahead and erase this business too. That stuff's in your notes. You have the beauty of pause on video, don't you? So you can make sure that you get this stuff right. Unlike in class when I'm rolling along. All right. Excellent. Okay, so this is our goal today. And really what I want to do with this transport concept then is create this section crossing membranes. So how do substances cross membranes? This is in the same chapter that we're in. This is chapter 7. And what we want to do is kind of lay this out into several obvious categories. So we want to look at passive versus active. This is going to be something that we have to address. It's almost like an outline that I'm going to give here. We also want to talk about polar versus non-polar substances. And we also want to talk about small versus large items, substances / items. So this is the whole rest of the lecture, really, is to break this thing down. So the first thing that we're going to do, this one has to do with energy. This one has to do, I'll underline these, passive versus active, polar versus non-polar. This one has to do with polarity. This one has to do with size. We're going to look at crossing membranes from those 3 perspectives. Okay. Let's do a little erasing here. All right, so the first thing I'm going to do is kind of address passive. So we'll take energy down first from an energy perspective. The word passive means no energy required to make it happen. Then that's going to be versus active. Active is going to require energy. And the energy, this is again something for later, but the energy of cells that we typically talk about is ATP. And I guess I used orange for this. You're going to see in your book that ATP has this kind of comic book POW sign on it. And I will honor that as well. Comic book POW for cellular energy right there. So ATP is what it's going to require. We're going to spend a nice set of lectures talking about how ATP is made in cells. But right now, passive, no energy, no ATP required. Active, energy is required. All right, so we'll see how that works in a minute. The other one we want to look at is this issue of polarity. Polarity is kind of, the polarity of a substance has tremendous impact on the, what I'm going to call the route across a membrane. How something goes, what pathway. So the first type is substances that can go directly through the membrane. So non-polar molecules, and these aren't large molecules. I'll say generally small ones. Non-polar molecules can actually go directly through the membrane. So they can cross that hydrophobic part of the phospholipid bilayer directly through the membrane. Examples of substances that do this, non-polar substances, small hydrocarbons. You'll remember what hydrocarbons are. You remember our definition. So hydrocarbons are molecules made pretty much of carbon and hydrogen. And of course that means they don't have polar covalent bonds. Of course that means that they're non-polar. Other ones, of course, look at this guy, O2. So O2, there's the molecular formula. O2 is a substance that's completely non-polar. And in addition to that, this guy, CO2. So here's another one, another molecule, linear, non-polar. So carbon dioxide doesn't have any great polarity, and O2 does because of the linear nature of it. Okay, so these guys can get through. The other category here is under polarity is going to be how polar molecules get through. Polar molecules, and again we're talking about smaller molecules, polar molecules. I'm going to put in parentheses small here. And in addition to polar molecules, we call them atomic ions. So what I mean by atomic ions here is that atomic ions are atoms, so cations or anions. We're not talking about molecules themselves that have ionized. So atomic ions and polar molecules, two things. How do they get across molecules, across membranes? These guys have to go through proteins. So they need the help of proteins. And we call these proteins transport proteins, which we've already done, but I'm going to do it again. So this is redundant. We're going back to what I've already told you. Proteins. So I'm going to have another category in the outline here for this business. So they need the help of transport proteins. And the transport proteins in question, the first of these, I'll use like a little I in my outline, would be the channel proteins. We've already drawn this. See drawing. And then we have, in addition to that, carrier proteins. So we can relate this to energy as well. Carrier proteins, channel proteins are open, and as I mentioned, these require shape change. They require shape change. So again, I want to refer you to the drawings that we made. So here we go again, channel proteins and carrier proteins. So I think that we're ready to move to this last category. And, well, no, before we do that, we're saving size. Now we got to plug in some really cool information. This is going to have to do with the direction with which these processes occur. So I'm going to start and get a whole board here. All right, so let's take a look at this. So there's some processes that kind of govern the way that things work. We have to go back to this is some background. It's kind of the rules of crossing the membrane. So the first thing we want to do is refresh ourselves with mixture terms. We've done this before. I already did this with water, but I want to do it again to bring this right back again. So remember that we had the solvent, which is the major component of a mixture. And of course, in biology, in life, it's always H2O, your friend that you know so well. Then we have solutes, and the solvents are the minor components. I've already identified some solute categories, but most of the things that we're talking about, you know, nonpolar, small molecules, that's a type of solute. And then we would have underneath that polar things, and that could be small molecules. And you can also have, as I mentioned, atomic ions. So these are going to be solutes. Whoops, atomic ions. These are going to be solutes in the game of crossing membranes. So there's our mixture terms. So then the processes, we've got two processes. The processes here would be diffusion. We're going to define diffusion, and then I'm going to show you a little picture of it. So diffusion is going to be defined as solutes move from areas of high solute concentration, I'll say high concentration, to low concentration. Okay, let's do an experiment. Experiment for diffusion. Got a beaker, and in the beaker we have H2O. So here's our H2O, and we go ahead and drop into our H2O some red dye. So here's our little pipette with a dropper, and inside of the dropper, whoops, wrong fit, is red dye. Out comes a droplet. So this droplet will hit the water right off the bat at T equals 0. And then basically we're going to watch this thing through the course of time. We're going to watch this thing ever so slowly. We're not mixing this. This is just happening. We're going to watch this thing kind of spread out a little bit. You may have done an experiment like this before, where it's kind of spreading out, starting to again go from high areas of concentration. It was super high red dye right in the middle of it when we dropped it in, and 0 here. Now it's starting to diffuse and spread out to the point where so this is T equals later, and then this is going to be T equals later again in the beaker. So now we will have, when it's reached equilibrium, we're going to have this whole beaker of water be a uniform pink color. So this is T equals equilibrium. Oops. So T equals, later yet. So this stage here is when the whole thing is going to be at an equilibrium, where the concentration of the solute molecules in any given place are equal. So that's diffusion. Take a look at diffusion in all of its beauty, and that's the first thing. Now, I want to remind you, very importantly, that this is passive. I want to remind you pretty much that what we're talking about here is passive processes. So we've added the energy component to it as well. All right, so that's that one. Now we've got to keep this party going and do the other one. It's great you guys have done a lab on this one already, so you should be very, very solid with these processes. There's diffusion. Here comes there's diffusion, and here comes osmosis. What we have to do for osmosis is introduce a new concept. So the new to the mix is what we call the semi, aka selectively permeable membrane. Now, just to let you know, cell membranes, plasma membranes are this. Plasma membranes are this. I've just spent the last 30 minutes talking about it, really. Are selectively permeable. So I want to underline those things. So the selectively or semi-permeable membrane. So the classic experiment that you can look at in your book, so you can see in your book, which I don't know the page number right now, but the classic YouTube experiment. I'm not going to draw the YouTube experiment. I'm going to show you a different one, just to show you the same process. So in mine, the book has the YouTube experiment to show it. I think it's really good for you to look at that. But I will also show you a beaker experiment. And in my beaker experiment, we're going to take a beaker of water, like we always do. Let's define it, by the way. So osmosis definition is going to be that solvent moves across a semi-permeable membrane from areas of low solute concentration. This is a big definition here. To areas of high solute concentration. Okay, so we'll take a peek at this one. I'm going to get myself, for this experiment, an intestinal casing. So this could be like a sausage casing, which is the epithelium of intestines of animals. Of course, we shove a bunch of meat and spices in there and call it good eating. So what we're going to do in the sausage casing is we're going to fill it with a red dye in water. So here we have pure H2O. And then in this experiment, we've got red dye. And of course, the red dye, that's the solute in this case. Red dye is the solute, and H2O is the solvent. So what we're going to do is we're going to plop this thing in to the beaker. And of course, the H2O level will go up in the beaker because we've added volume. And here's what's going to happen to our thing. So we've dumped it in the beaker. Here's what it looks like at the beginning. So we still have this nice concentration of red dye. So we'll say this is T equals 0 down here. T equals 0. And then later on, we're going to come back and take a look at this thing, and we're going to see this. So there's a little bit of flex in our sausage casing, and that thing is going to be stretched out to the max. It's going to be stretched out to the max, and the red is going to be, of course, less intense because H2O rushed into that. Do you see what's happened there? Now there's a limitation. This is an equilibrium here. No more water can get there because this thing is as stretched as it possibly can be. So that is osmosis. So what we have, going back to the definition, the semi-permeable membrane is the sausage casing. This thing right here is the sausage casing. We've got solute, which can't cross the membrane. The red dye that's in here can't leak out of it. It's stuck in there, but water can. So we dump this thing down into water, and water's what crosses the membrane. And water's going to go from an area of low solute concentration, and it is radically low it's radically low right here. That's just pure water. And then inside here, the solute concentration is quite high. So it's going to go from low solute to high solute concentration. That means it's going to be rushing in to the sausage casing, crossing the membrane to tend to an equilibrium. So there you have it. Check this out, osmosis. Go and take a look at the old YouTube experiments too. It's pretty cool. I'd like you to be able to interpret those, but that's in your book, so I'll make that as a special assignment. Sorry, I don't know the page numbers. Didn't bring a hardbound book with me, and the other one's in the same computer that's recording this lecture. All right. So I think we're doing great. So now we have these ways that things can get in. Oh, oops. Okay. The other thing to say, osmosis is also passive. There's no energy that has been spent here. No ATP whatsoever. This thing is passive. So we got diffusion and osmosis that set rules for how things can get across membranes. So if we go back and take a look at this, we have the things that go directly through membranes, they diffuse. Then we have things that can't go directly through membranes and they have to go through transport proteins, of which we have 2 types. So that's that. Take this information one step further and we will see how many things get across the membrane. Okay, here we go. So, next part of our story here is that, let's see, we can put the whole thing together now. Okay. So, now we can take all of this, add it up, and talk about specific ways across membranes. So, simple diffusion, the first thing, crossing membranes. So, simple diffusion of nonpolar substances, of course, remind ourselves, passive, nonpolar substances. The second one, checking the list, this is a new term to add to our lecture, facilitated diffusion. So this is the catch-all term. Facilitated diffusion is where certain hydrophilic substances go through transport proteins. Okay, so we can take a look at this. The transport proteins that we have, I'll draw these again, but of course we have channel proteins. And this one we could draw, put a little bit of phospholipid bilayer here, and draw this as the big donut that we drew before. So that's that one. Here's a nice solute traveling right through that. The next one that we have is going to be those through carrier proteins. And in the carrier proteins, you've got 2 different kinds we'll talk about. The first one, ion channels. So ion channels are carrier proteins that transport ions. All right. So we've got that going for us, and I want to add to this list kind of a second little category. Maybe I'll throw an A here. My outline scheme is kind of strange letter-wise, but it should work. And I want to say, note that some ion channels are what we call gated channels. Now, my best explanation for what a gated channel is it's kind of like the turnstiles that you go through when you go into a big stadium, like in Sports Authority. You know, it's one person at a time, and those things only click so fast. So you're going through this line, and you know that you could go a lot faster if there was not a gate there, but there's this little thing that rotates and clicks and stops, then rotates and clicks and stops, rotates and clicks and stops. A gated channel is like that. If you looked at the picture of a gated channel, it, like other things that we've talked about, requires a shape change. Remember though, this whole thing, facilitated diffusion, simple diffusion, everything on this board is passive. Everything on that board is passive. Requires a shape change. So this is where you have something, you know, some kind of a transport protein that might look like this. And here it is in its open position. Let's get some phospholipid action here. So here's a position where a solute could bind to it and fill that space. And then the next position for the same protein is where the whole thing is. Let's see. I seem to have lost a pen somewhere. Oh, there it is. So this next one is like this. So now the thing opens the other way, and the solvent, or excuse me, the solute flies out like so. Let's get a few phospholipids here. So that's the whole thing. You can see that this thing is open this way here, and then it doesn't work very well in the light, but so it's open this way, and then it inverts and opens to the other side. So substances can get into the turnstile and then out. Into the turnstile, out, in, out. Clicks. So when this thing has a large number of solutes on the other side, it only goes so fast. And so there's a rate concept right here. Because of this, this gated channels, there is in gated channels going to be a rate. They're going to be limited. They're going to be rate limited. There's going to be a possibility that they can only go so fast, maximum, just like right before a football game. Everybody's lined up and they can only go in so fast. They can't go as fast as they could do if the gate wasn't there. So this is a type of ion channel. So ions do this, but this is a type of ion channel that actually requires a shape change. That's excellent. All right, so there's carrier proteins. The next thing for us to do, we're making great progress. All right, here we go. So the next thing for us to do is to take a look at active methods. So there's really just one thing we're going to do, active methods on this page, and that's going to be pumping. So pumping is going to require carrier proteins or pumps are carrier proteins that require the use of energy, that require energy in the form of ATP. So what I mean by that, ATP, and of course, as I said, every time we do that, we've got to throw superhero pow on it. That's cellular energy. It's a very, very important player for much of our class moving forward. So this pumping process is going to require carrier proteins that require this energy. And what we're going to do is go straight to the example in the book of the sodium-potassium pump. I'm going to use the symbols because you guys are so hip to symbols. This the Na plus K plus pump, a very famous pump or carrier protein that requires energy. So I'm going to try to draw this one a little bit more streamlined. How about that being a phospholipid bilayer? And let's use a pump with this color. We're going to make the left side with 3 little circles. So make sure that when you draw this, you've got 3 semicircles there. That's going to be the side that represents the binding of sodium. And we're going to show it this way with 2 small openings like your book does. That's going to be the side that binds potassium, and it is going to have just two slots. We're going to make a legend for our substances. And let's see, these colors could end up being backwards from the way that they are in the book, but I'm going to use a blue circle to represent Na plus, and I'm going to use a green square to represent K plus. So we're going to start this thing with lots of sodium out here. And we're going to have a little bit of sodium down here, and then we're going to have a little bit of potassium out here, and we're going to have a lot of potassium in here. So I want everybody here to make sure that when you see this particular picture, we know that at this particular point in time, here we have a high concentration of Na plus and a low concentration of K plus. You're saying, what's up with the brackets? We've done this before. I want to remind you that shorthand for concentration of substance X is equal to brackets around X. That's the universal shorthand. You'll do this day in and day out in chemistry, and that's what we've got going there. So notice that I've gone to high sodium and low potassium, and of course the flip side is true on this side of the membrane. I'm going to go ahead and draw my phospholipid bilayer. Keep in mind that I've made this much more cartoonistic. I'm getting tired of drawing circles and bars. So that's what this thing is. So down on this side of the membrane, which is intracellular, so this is intracellular. What I'm going to do here is say that we have high K plus and low Na plus. Now, that's the last time that I'm going to do this in the picture. I'm not going to make as many frames as your textbook does. So we want to go to the next step. And in the next step, what's going to happen here is that we're going to have Na plus binds. So draw this one this way. Get the gate. Okay, Get that going on. So we're going to have a couple of things happen here. We're going to have sodium molecules that are going to actually come up and bind, even though the concentration is very low. So these molecules are going to come up and bind. And then I'll draw a whole bunch of our K pluses here just to remind you that we have got tons of K plus on this side. And then not much K plus out here. And of course, tons of sodium out here. So sodium is binding. Sodium is binding right here to the pump. This is going to make the next extremely cool thing happen. So ATP is going to become ADP. And it's going to do so by giving a phosphate, by binding a phosphate. I'm going to change that. Okay. So it's going to give a phosphate that will bind physically to the pump. So that's super, super important there. I don't know if you can see that that's a P or not. I hope you can. Let me modify this. That's a P. That is a phosphate right there. Right there. So this particular part right here, what's happened there is what we call a phosphorylation. So it's called a phosphorylation. That is what has happened there. Now, I want you guys to remember this then. We've talked about the impact of having something so incredibly charged show up close to other substances that are either charged or not charged. You're going to have all of those interactions. When this thing gets a phosphate group on it, it causes a shape change. So shape change happens from this particular point on, and that's what I'm going to draw now. Now the shape change for this guy is to open up the other way. So there's that side. There's this side. And notice that I've taken these guys like your book does and opened them up. So here they were kind of pinched together, these little V-shaped notches. And here when it shape changes, they open up. These guys kind of scrunch together, which is what I'm trying to show, because what's happened here when it opens is out goes, these guys go and further the concentration problem on this side of the membrane. Keep that here. And then when this happens, the phosphate group leaves. So here's the P group, and when it leaves, you call it PI for inorganic phosphate. So this guy leaves. Let's get some of the rest of these guys down here just to remind you that everything is the same as it was. So we have a large concentration of potassium here and a small concentration of sodium. Up here, a large concentration of sodium and a small concentration of potassium. But what I'm going to show you is that the potassium ions come in and fill their spaces at this point. So I've stuck a couple of steps together here compared to the book. Your book will do this in 6 steps. I'm doing it in 4. So now you have potassium binding to the receptors after the shape change. And then the next step over here is going to be when the phosphate leaves, the shape change again. So another shape change back to the original. So here we have that. Phospholipid bilayer, and at this particular point, the potassiums go into their place, which of course is full of potassiums, but that's the key, pumping against concentration gradient. That is exactly what we're talking about here. We are talking about a pump. It takes energy to go against the concentration gradient. It doesn't take energy to go down it. So let's reestablish that. We've got tons of this material here. Of course, the same thing's going to happen here. These guys are going to start to go up and fill, and the whole thing just kind of keeps its cycle going. So here's a 4-frame view of the sodium-potassium pump. Are you guys ready for the key for this? The key for this is to understand that when ATP, which the T stands for tri, becomes ADP, T equals tri D equals di. So when the when the ATP loses a phosphate, it gives this phosphate to this massive transmembrane protein, this carrier protein. It sticks it, it phosphorylates it. It binds physically to the big protein. And it's such a hot potato that the charge interactions cause this whole thing to just change its shape. So the cartoon shows it opening up the other way, and it's at that time that it's exposing its potassium receptors to potassium, and it's dumping its sodium against the concentration gradient. Then when the potassium guys come in, they cause the booting off of the phosphate, and now that it doesn't have a phosphate stuck to it, it changes back. It flips right back to its position that it normally occupies when it's not phosphorylated. And then, of course, that means that sodium can bind, potassium can leave, and the whole thing cycles and cycles and cycles. These are extraordinarily important pumps in your body. Look up a nerve cell, look up a neuron, and you're going to see sodium-potassium pump, sodium-potassium pump, sodium-potassium pump. This is hot stuff. This is it. That is amazing to me. Man. Okay. So, we're doing incredibly well because I want to hold this to standard lecture time. I have 7 or 8 minutes left according to what I see, and that means I can erase and have another go. What we want to do now is look at big stuff. That's small stuff. All right, here we go, last dance. So now we're going to talk about big stuff. Well, the big stuff gets into cells from outside by what we call endocytosis. There's 3 types. One is what we call pinocytosis. Pinocytosis is where smaller macromolecules get into the cell. We're going to draw a whole cell. So here's an animal cell with a double phospholipid membrane right here. Might as well give you a nucleus, but that's about as far as I'm going. So there's our nuke right here, nuclear envelope. This is the plasma membrane. So that means this stuff is cytoplasm. I'm not going to draw any organelles. What I want to show you, though, is that we have solutes. So these are going to be these smaller macromolecules right here. And we're going to show the pinocytosis process. I'm going to blow it up. So here's the membrane looking like this. Here's the molecules. And what we will see, we're going to describe just how in the world that this can happen. We're going to see the thing kind of just form a little pocket. And in that pocket, of course, we're going to have solutes. And that process will continue until the whole thing just dramatically pinches off inside of the cell. This will form a vesicle. So, new term here, a vesicle. And a vesicle is a membrane-bound compartment. That's that. The next type is called receptor-mediated endocytosis. So this one has a specificity to it. So here we have our cell, and the cell's going to have receptors on it. And we'll have a solute. So our little substances here will bind to the solute. And when that particular process happens, we're going to stimulate the same kind of process where the membrane pinches around. And ultimately forms a complete vesicle here. The difference between this vesicle and the previous one, the difference between this vesicle and the previous one is that, of course, we have our little receptors that have bound their substrates. So that's that. So the thing about this is that this guy has a specificity. It binds specific things, whereas this is general. General small stuff is the idea here, and specific substances that bind specifically to receptors. And the receptors themselves are proteins. Finally, the last thing is what I promised to talk about. The last of these endocytosis processes is called phagocytosis. I referred to this in the beginning of the lecture, so here it is. Phagocytosis. So this is large scale kinds of processes. The classic example of phagocytosis that I'll give you is a white blood cell. I've already talked about white blood cells, and here's what a white blood cell looks like. The difference between a white blood cell is and other cells is that they are kind of irregular and dynamic in shape, and they call these little outcroppings here, these little bumps, pseudopodia. Interestingly, if you took a look at a white blood cell, you'd see that the nucleus was a really wild-looking thing. The nucleus might look something like this. They have what's called a polymorphonucleus. The you guys don't need to know that right now. I just wanted to show you that if you took a look at a white blood cell and you tried to relate it to what you know about basic cells, this thing's not going to look the same. So here's the thing. Let's say that these guys here are bacteria. So these are bacteria, invading bacteria in your system, and they have to be dealt with. The process here is very dynamic, but in short, let's draw this cell changing its shape and kind of moving to gobble up this business. I'm trying to draw a phospholipid bilayer here. I can show you that the nucleus also changes. It's like the nucleus of these things is like a lava lamp itself. And then let's put some of these bacteria here, and the next part of the process is the whole thing is gobbled up. So here's my white blood cell. Here is a new vesicle here that has formed, that has bacteria in it. And then let's draw the nucleus just to show that it's still intact and awesome as it always was. So this thing is called a phagosome. That is the process that is a vesicle called a phagosome. It's also known as AKA a food vacuole. A food vacuole. Good grief, I'm going to have to do that again. Food vacuole. Excellent. So this is phagocytosis. It's large scale. This is what white blood cells do when they gobble up invading things like bacteria. And it's an amazing and dynamic process. What we're going to do when we talk about the cytoskeleton is I'm going to show you that inside of cells like this incredible crisscrossing network of massive proteinaceous filaments. And these filaments are capable of crawling relative to one another. And that cytoskeleton is what enables a cell like this to change its shape so dramatically. I mean, shape change is huge in biology. Think about muscles. Shape change. Okay, so that is my time. I've actually run 3 minutes and 20 seconds over. Apologize for that. I am going to try to get this thing online so you can have a look. And I want you to definitely get this thing under your belt before we meet on Thursday. It's August 6th now. And Thursday is going to be what? August 9th. So do this thing and show up ready to keep going on class on Thursday. Excellent. Ciao.